can I just maybe, can I switch to my laptop? I'm sorry. It's not only he was late, but he basically had no idea how to click. He messed up a computer. I am truly sorry. Let's do something else. Oh, so now it's moving. So do we give it a chance? Yeah. Okay. Let's try. Okay. So you said this. You're a man with the golden tongue. Okay, very good. So this is a quick, very simple example of weakly coupled linear system in which I actually have the ability to solve the underlying equations. This was one of our papers a couple of years ago about so-called retrotransposition systems, which are this sort of interesting system because people think that one way in which evolution marches through the genomes of the species is by doing this random mutation. One way in which we could try to model random mutations is by means of these retrotransposition genes. But I don't really want to talk about this right now. I just want to say that let's suppose I have a very simple system. And I will take my classical chemistry now and I will try to apply it to a biochemical. Okay, so in fact I will try to apply it to a genetic model. So let's suppose that I have a genetic DNA somewhere here inside a cell. And then I'm looking at the polymerase, which is moving along. It creates a copy of the transcript of the appropriate gene. Now, this polymerase will go faster or slower depending on this additional molecule here, which are called T-prot. This is sort of a facilitating protein. It's not the protein that's translated from that gene. It's just some other protein that speeds up or slows down the rate at which my transcription process moves forward. So basically I have two chemical species. I only care about those two. I have a transcript and I have a protein. And the first reaction basically tells me that this protein is really a catalyst. It works, it doesn't get consumed in this reaction, it doesn't get created, it basically stays put. But the rate, because I'm following the law of mass function, the rate of this reaction will now depend on how many of these T-prots do I have available. And the other thing which is important is these T-prots I will only care about when they are here. In other words, when they are on the genomic DNA, because then that's where they can react with the polymerase. If they are not on the genomic DNA, then I don't care about them. There might be very many of them somewhere out there, but they are not in the system, they cannot react. So basically, I don't keep track of those. I only keep track of the ones which are on the polymerase. And the other thing is that I just told you, I keep track of these things on the polymerase, so I have just violated my assumption of uniform mixing, right? So the system that I have just described on the previous slide is not necessarily the system that's exactly described in the reaction. But it turns out that you can actually work out that the error is not very large. So there is some sense in making that uniform mixing assumption, partially because this entire system floats around. And again, the error that we're making by assuming uniform mixing is smaller than a whole bunch of other errors that are related to, for instance, measuring these systems. So as a sort of first approximation model, it seems to be working okay. At least it did in some of the experiments that we have worked on. So basically now, this is just an example. It's a very simple system. It has 4 chemical reactions. It has 2 species. The first reaction says that I'm creating these T-prots at the rate which is proportional, I'm creating the mRNA at the rate which is proportional to the amount of T-prots. Then the mRNA either gets translated or gets scrapped or gets lost, I don't care. It basically gets out of the system. And then I have these T-prots which are created out of thin air, but that's what I just said. They really aren't, because I am just trying to see the T-prots which are on the genomic DNA, so whenever they get off, then they are somewhere floating around, but they're not in my system. So that basically means that I'm recruiting some number of these T-prot molecules on the genomic DNA, and then the fourth reaction says that, well, the T-prots could get out of the genome, right? So they can basically return to the cell.
What is phi?
Phi. Oh, this is not phi, I'm sorry. This is an empty set. That's how, so it basically means that represents a loss of a molecule. Okay? So it basically means that there was a molecule of mRNA and it got annihilated. This basically means that there was no molecules and the molecule was created. Again, this is against the conservation laws and a whole bunch of laws of physics. But you have to understand that this is in a very specific context. Okay.
So you first, you're looking at creation of mRNA, right? So why do you have T-prod?
Well, the idea is that this T-prod is actually the factor that's regulating the rate. Okay. And the idea, again, I don't want to go into this too deeply, but perhaps this is interesting for you. So I would be happy to answer. The idea is this: that this L1 gene, which is basically a retrotransposition gene, is a very unnatural being because it attempts to make a copy of itself. It transcribes itself, but then instead of translating, it actually attempts to reinsert itself in a different position in the genomic DNA. Now, the cell typically does not allow, or it tries not to allow this to happen. So in particular, healthy cells will have mechanisms that will attempt to slow down this process of transcription. However, if you introduce some kind of external stimulus that will disrupt some of these defensive mechanisms, the cells will actually give up and they will allow for these reactions to happen much more frequently. The way that you can model this is precisely by adding this product. Because this is an attempt to sort of modulate the speed of transcription. And yes, you're right, in general transcription is a deterministic, it's a deterministic phenomenon. But in this setting, you are thinking about it as the ability of the polymerase to move faster or slower because basically some of these polymerases actually will be more successful. Okay? Typically what happens, polymerase attempts to make a transcript and then it basically gets kicked out. This transcript is not finished, it will be scrubbed. Okay? So in general it means that the process of creating full transcripts is slowed down. Now if this kick out mechanism doesn't work, then this sort of, you can see it by increasing the number of molecules per units of time, so you can kind of model it at this level. I mean, we have a paper about this a couple of years ago. You can look it up. It describes the whole business. Here I'm just using this thing as an example of this chemical reaction, and in fact the talk is not really about the chemical models of this kind.
Sorry, let me ask you, if K3 equals K4, you just can remove two equations?
So this is decoupled, right?
Yeah, so for K3 equals K4, you'll get...
Yeah, I mean, this is completely decoupled. This is like a birth and death process with the same rates, right?
Yeah, so just...
Yeah. The nice thing about it is that you can solve this as an ODE model. But stochastically, you can't. In fact, it's an interesting exercise in trying to write out the generator of this chain, because it's a trivial equation in terms of an ODE model, but in terms of the stochastic generator, it's actually not very nice.
It's difficult to write.
You cannot write you cannot solve Kolmogorov equations, and you cannot easily identify the closed form of generators, even by partial differential equation. And then you really can solve it. I tried for a long time. Okay, so basically, so this is a nice system. It's very simple, but on the stochastic side, it's actually not computable. Okay, so it also tells you how difficult these stochastic models are. Let me just skip over a little bit. So what happens? In this system that I just showed you, how do I get to the deterministic approximation? Okay, I am rescaling my system into these concentrations C1 and C2. And then basically I write my integral equations and then I differentiate. So this is a differential equation. This differential equation is a deterministic approximation of the behavior of that system that I just showed you on the previous slide. In some sense, this stochastic system is attempting to track that deterministic system with some fluctuation. And there is a result that says that basically this fluctuation is Gaussian with a very complicated covariance structure because this is a Markov process. Okay, so this is the result from 1968 by Professor